So over time, histones are not returned to the right places, and DNA methylation is added in places where it shouldn't be.
We can read that methylation pattern, and I could tell you how old you are, exactly, and when you're even going to die.
How could you tell that?
Well, it's a clock. We call it the Horvath clock, named after my good friend Steve Horvath. And so these little chemicals that accumulate on the DNA, like plaque on the teeth, we can read that, and the more you have, the older you are biologically. So you might only be 40. You're younger than 40, of course, but I'm 50 now, but I might be biologically 60. Actually, I was, and I changed my life, and then the test said I was biologically 31.
I mean, one of the things I found really interesting was you found a way to make mice age faster. So how'd you do that?
Well, the clock of aging is due to the loss of the information in the cell. And one way to accelerate that is to go break a chromosome. Instead of going in the sun, we engineered a mouse where we could break its chromosomes. Not enough to cause mutations. The cells put the DNA back together, so we didn't lose any genetic information. But if we're right about the epigenetic information theory of aging, those mice should get old. And that's exactly what happened. It's gray, it's got a hunchback, it's got dementia, all its organs look old. But the real test was, what if we measured that DNA clock, what we call the DNA methylation clock? And we measured it, and those mice were actually 50% older than mice that we didn't treat. So that isn't just a mouse that looks old, that mouse literally is older.
What's interesting about this hypothesis is that if it's true, if the noise accumulating in the epigenome is really what's causing aging, well then there are steps we can take right now to slow the rate of aging in our bodies by trying to better maintain our epigenomes. So how do we do that? There's this theory that billions of years ago, early bacteria took an important evolutionary step. They actually developed two different modes of living. When times were good, they used their energy to grow and reproduce. But when conditions were tough, they used their energy to protect and repair their cells. They evolved what Professor Sinclair calls longevity genes. These genes, triggered by adversity, create enzymes which, among other things, maintain the epigenome. And today, those same longevity genes can be found in bacteria and us.
We have these hormetic response genes, or longevity genes, that are in all of our cells. And they sense when we've run a lot, we've lost our breath, or we're hungry, or a little bit hot, a little bit cold, that these genes are turning on our general defenses against aging. So what is that? So parts of our cells fall apart, they can put them back together. Proteins misfold, they can get rid of them or put them back together. The ends of the chromosomes get shorter, they can lengthen them. A lot of processes that go on, but one of the most important, I think, is maintaining the information, the epigenetic information in the cell, so that our cells don't forget what to do. There are three types of longevity gene. They're the ones we work on, called sirtuins, and they control the information in the cell. In fact, SIR in the sirtuin stands for silent information regulator, number 2. There are other ones. The other group is called AMP kinase, or AMPK. This group of genes senses how much energy we're taking in, mostly in the form of sugar. And then the third group is called mTOR, and these genes control and respond to how much amino acids we're taking in. So if you eat a giant steak, you've got a lot of amino acids coming into your body. That'll actually prevent mTOR from hunkering down and keeping you being longer lived. So the mouse experiments actually bear this out. The best way to make a mouse live longer is to reduce the amount of time it eats, so periodic fasting, intermittent fasting, to keep it a little bit cool, and to restrict its amino acids. That's the recipe for long life for a mouse. And it's true for monkeys as well. There have been calorie-restricted studies where these monkeys for 15 years didn't eat as much food as the ones that gorged themselves whenever they wanted. And they were protected. They didn't just age slower, they didn't get as much diabetes and heart disease. They were actually fit and healthy when the control group, eating whatever they wanted, aged and became sick quicker.
When some people think about eating less, like calorie restriction, as a way to extend their life, that doesn't seem like a very pleasant way to extend life. I mean, to be hungry for longer. So are there other ways to, mimic that effect or to simulate that?
There are these molecules that turn on the sirtuin pathway and trick the body. And so, for example, in the lab, if I give some of our mice a molecule called NMN, which raises the level of a chemical called NAD. You get hyperactive defenses in the body.
And what did you see in these, senescent mice that you gave NMN to?
Well, we had a bit of an incident. These mice that we gave NMN to ran 50% further, but actually some of them ran so far that the machine, the little treadmill, stopped working. And we had to reprogram the software because this program had never seen a mouse that ran more than 3 kilometers.
3 kilometers for a mouse.
For an old mouse, they outran the young mice.
And that's like an ultra marathon for us.
That would be probably like taking a 70-year-old and making them run faster than a 20-year-old further. So these are ultra marathoners. And if we did that to humans, imagine you could have 90-year-olds winning Olympic medals.